How to identify AEM, CEM, and BPM membranes using dye tests. In electrodialysis and bipolar electrodialysis systems, correct membrane identification and orientation are critical for performance and reliability. A simple laboratory test using two common dyes, methylene blue and methyl orange, allows us to quickly distinguish between anion exchange membranes, cation exchange membranes, and bipolar membranes. This method is fast, low cost, and highly visual, making it ideal for laboratory work, training, and quality control. The test is based on electrostatic interactions. Methylene blue is a cationic dye. It is strongly attracted to negatively charged surfaces such as cation exchange membranes or CEM. Methyl orange is an anionic dye at neutral pH. It selectively binds to positively charged surfaces such as anion exchange membranes or AEM. A bipolar membrane or BPM contains both layers, one CEM side and one AEM side, each reacting differently to the dyes. The test requires only basic laboratory materials, prepared dye solutions, deionized water, simple containers, and standard handling tools. For the methylene blue working solution, prepare a 1 gram per liter stock. Dissolve 0.10 grams methylene blue in 100 milliliters deionized water. Dilute 10 milliliters stock plus 90 milliliters deionized water to obtain a 100 milligrams per liter working solution. For the methyl orange working solution, prepare a 1 gram per liter stock similarly. Dilute to 100 milligrams per liter and adjust pH to 6 to 7 using sodium bicarbonate or phosphate buffer to keep the dye in its anionic form. Before testing, the membrane is rinsed to remove protective liquids and soaked in deionized water to ensure full wetting. For bipolar membranes, marking one corner helps track orientation during the test. Method A. Separate immersion. 2. Small membrane samples are immersed separately, one in methylene blue and one in methyl orange. After 10 to 20 seconds, the membranes are gently rinsed and visually inspected. A strong blue coloration indicates a CEM. A strong orange coloration indicates an AEM. Mixed or weak coloration may suggest a bipolar membrane or a coated surface. Dual dye orientation test. To identify BPM orientation, the membrane is laid flat. Methylene blue is applied to one side, methyl orange to the other. After staining and rinsing, the blue side corresponds to the CEM layer and the orange side corresponds to the AEM layer. This method provides immediate and clear orientation feedback. For further confirmation, a simple diffusion or electrodialysis test can be performed. Assemble a 2-compartment cell separated by the membrane. Fill one side with dye and the other with deionized water. Observe color migration. Methylene blue passes through CEM. Methyl orange passes through AEM. Adding 1 to 2 volts DC can enhance visualization. Results summary. CEM membranes stain blue. AEM membranes stain orange. Bipolar membranes show 2 distinct sides, one blue and one orange. Notes and tips. Maintain pH 6 to 7 for methyl orange. Extend staining time to 30 to 60 seconds for coated or dense membranes. Ensure membranes are fully wetted before dyeing. Rinse after observation to avoid permanent stains. Use gloves. Dyes can stain hands and lab surfaces. The methylene blue and methyl orange dye test is a practical and reliable method for identifying and orienting ion exchange membranes. It is widely used in laboratory practice and supports accurate preparation of dissolved electrodialysis and EDBM systems developed by YASAET.